Dear students, welcome to the online classes of animal biotechnology. Today I will explain about the Human Genome Project, that is HGP, and ethical, legal and social issue related to Human Genome Project. So first, I would like to explain what is human genome and how human genome was sequenced. So in this video, I will explain how human genome was sequenced. So, as you know, the each individual is has having the specific genome. So we have bit different in our genome. So as you see in this figure, in all the cells contain the nucleus, and inside the nucleus there is a chromosome, and chromosome are made up of specific DNA fragments. Here you can see the chromosome and DNA, and each DNA the our genetic information are stored in the four letters that are A, T, G, and C. So these are the nucleotides. So most of our cells have two copy of genome. One copy is from the paternal and another is maternal. Uh, so uh, so each chromosome has the many letters of the nucleotide. So here you can see the length of the chromosome, and if you will see all the chromosomes, so our uh, all 23 pair of chromosome contain around 300 crore of DNA letters. So there are many countries were involved in the Human Genome Project. Like here, you can see the Wellcome Trust has sequenced some chromosomes. So the chromosome actually cannot sequenced as whole. So it has to be cut in the small or tiny pieces first. Here you can see this fragment of chromosome, and it is cut into small pieces. Further, one small piece can cut into another tiny pieces, and at the end, these tiny pieces were then sequenced. And here you can see the small segments. So that information built up to finally give the researcher the complete sequence of entire chromosome. So for the genome sequencing, individual or healthy individual were, or you can say, and these individuals are volunteers. So blood sample was collected from the volunteers, and the DNA was extracted from the blood, and it was cut or broken into small pieces. And these small pieces were then cloned in the bacterial plasmid. Here you can see the plasmid, and this plasmid is then transformed into the bacteria for the multiplication. So all bacteria here can contain the human gene. So there are different centers around the world involved in this project. So each uh, institution was involved to sequence the different chromosomes. So the chromosome was cut into small pieces and cloned into the bacterial plasmid, and these transform into the bacteria, and these bacteria grow into the small colonies, and finally these colony were picked up by the robotic method. Here you can see how this robot is picking the colony, and it is putting into the sequencer in the small tubes. So here each small tubes contain different uh, genes from the human. So here you can say this is the bacterial part, the green one, and that pink one is the human gene, which is unknown sequence. This is known sequence, and this is unknown. So the sequencer works on the principle that is called Sanger-Didoxy method. Here you can see the normal cell. In normal PCR reaction, you can say that the polymerase can extend. This is a DNA polymerase, or tech DNA polymerase is a simple PCR reaction. It can extend, or it can add the complementary region of this strand in both strands. Here you can see how that DNA polymerase is adding the complementary sequence. But in the case of Sanger-Didoxy method, we use dideoxy nucleotides that are fluorophore labeled, and this Sanger-Didoxy sequencing depends on the chain termination. So because we use the DDP, that is dideoxy, so once this dideoxy, which is fluorophore labeled, comes into the extension chain, it get terminated. So you finally you can get many fragments of the DNA after the PCR reaction. So here it is more clear that when you denature the both fragments separates, and the second step, primer or anneal. So primer are designed on the basis of the sequence of the bacterial gene. So here these are the sequence of primer. Then polymerase come into the act and it extends the complementary sequence. And once there is a dideoxy or DATP comes, it get terminated. Similarly, when DDGTP comes, it get terminated, and similarly DDTTP, when it comes into the extension chain, it get terminated. So finally you will have many small fragments, like here you can see the different fragments of PCR products, and these PCR products actually get terminated at the different DD and TPs. Here you can see the different by different colors because they contain the fluorophores. So here you can see the what sequence it is in the this strand. So these small fragments are actually float in the liquid, and they can then run into electrophoresis, that is capillary electrophoresis, which is inside the sequencer. And in capillary electrophoresis there is a laser beam. So once these smaller fragments pass through the laser beam, they give the specific fluorescence, like you can see here. This yellow will give yellow fluorescence, then green color, then yellow color again, then blue, yellow. So all the nucleotide can get the different color. So finally we can get the sequence like this. This is a sequencing data. So actually the all colors actually have the different nucleotide, like T is red, G is yellow. So finally to rebuild the entire genome, all sequences are combined and searched for the overlapping. And here you can see the all sequences are uh, combined, and here you can see the overlaps. So all the raw data then finally assembled, and the sequence are made available freely online. Thus, the Human Genome Project completed. Hope you understood this. Now I will explain the in written that what is Human Genome Project. As you have seen, the Human Genome Project was an international scientific research project that aimed to determine the complete sequence of nucleotide base pairs that make up human DNA and all the gene it contains. And it is the world's largest collaborative biological project so far. Actually, the idea was picked in 1984 by US government, and it was launched in 1990 and completed in 2003. And the genome of any given individual is unique. Mapping the human genome involves sequencing the genomes of a small number of individuals and then assembling these together to get a complete sequence for each chromosome. So, as you have seen, the different institutions around the world were involved in the human genome project. So, each or every uh, research institution who were involved in genome sequencing, they sequenced the separate chromosome. Suppose Sanger Institute sequenced chromosome number 11. So the NIH coded for NIH sequenced the chromosome number 12. So different institution sequence the different chromosome of the human. So finally they were all assembled, and this finished human genome thus is a mosaic, not the representing any one individual. So the goals of Human Genome Project is to identify all genes in human DNA, to develop genetic linkage map of human genome, to obtain physical map of human genome, and further to develop technology for the management of human genome and information, to know the function of genes, and determine the sequence of 300 crore, that is 3 billion chemical base pair that make up human DNA, store the information in public databases, and develop tools for the data analysis and transfer related technologies to private sector. So these are the goals of Human Genome Project. So now I will discuss about the participating countries and funding agency. So it was started in 1990 in USA with the help of Department of Energy, that is DO, and National Institute of Health, NIH. So and then other universities around the world from different countries like UK, Japan, France, Germany, Canada, and China also involved in the Human Genome Project. And apart from these university, a private company, uh, Clara Corporation, was also involved in the Human Genome Project. So here I am discuss about the timeline or the breakthrough of Human Genome Project. So it was started in 1990, and James Watson was the pioneer project director of this project. You heard about the Watson, so he was the pioneer and joint project director of this Human Genome Project. And in 1990, first gene was mapped, that was BRC1, and that is breast cancer gene. It was sequenced first. In 1993, first 5-year plan for HGP was published, and then Sanger Institute from UK also joined the Human Genome Project. In 1994, uh, human genetic mapping goal was achieved. In 95, Genetic Privacy Act was passed, and then first bacterial genome was also sequenced in parallel. So in 1996, human gene was published. Uh, First human gene was published in Nature, and yeast genome was also sequenced. In 97, NIH actually became the National Human Genome Research Institute, and E. coli genome was sequenced in 97, parallel to human genome. Uh, in 1998, second 5-year plan of HGP was published, and then Japan, China, and other countries also involved in the Human Genome Project. In 1990, sequencing of human chromosome 22 was completed, and it was published in Nature. In 2000, working draft of human genome completed, and 2001, working draft of human genome sequence was published in the Nature. And finally, in 2003, human genome was finished, and finished version of human genome sequence was completed, and HGP ended with all goal achieved. Now the technical aspect, as I already discussed in the video, that there are two processes human genome sequencing. First is genetic mapping, and second is sequencing. So first, for the genetic mapping, you may heard about this technique, that is RFLP, restriction fragment length polymorphism. As you have seen in the video, the DNA can be first fragmented in smaller pieces. That is called mapping strategy, or gene mapping. And the second step is sequencing. Once we clone the smaller fragment in the desired vector, they can be sequenced by the Sanger deoxy method. So nowadays, all the advanced sequencer machines are based on the Sanger deoxy methods, and I have already discussed this Sanger deoxy method in the video. Now the outcome of the Human Genome Project, or the feature of human genome. So from the human genome, it was concluded that human genome consists information of 24 chromosomes, 22 autosomes, 1 X chromosome and Y chromosome. So the human genome contains 3 billion nucleotides. So that is 300 crore nucleotide. Human genome is estimated to about 30,000 genes. So there are 30,000 functional genes in human genome. So every gene consists of about 3,000 bases. So average size of each gene is 3 kb, but this size can vary from some bases to some million cases. And only about 3% of genome encodes amino acid. So only this 3% gene is having the known capacity, but the function of are unknown for over 50% of the discovered gene. The repetitive sequence make up very large portion of the human genome. And these repetitive sequences have no direct coding function, but they shed light on chromosome structure, dynamics, and evolution. So that maybe previously it was considered as a junk DNA, but now it is clear that they have some specific role in the chromosome structure, dynamics, and evolution. So chromosome 1 has the most genes. It contains almost 2960 genes, and chromosome Y has the lowest number of genes. It contains only 231 genes. Almost all nucleotide bases are exactly the same in all people. Genome sequence of different individuals differ uh, for less than 0.2% of base pair, and about 85% of all differences of DNA are due to the single nucleotide polymorphism. Now the ethical, legal and social issue, that is ELSI of Human Genome Project. So ethical concern of Human Genome projects are such as beneficence, do not harm, human rights, animal rights, authority, autonomy, ownership, justice, confidentiality or privacy, responsibility, scientists and social duty, and consequences. So, since the beginning of Human Genome Project, many ethical questions were raised, uh, like recognizing that Department of Energy and NIH allocated 3 to 5% of their total expenditure on the Human Genome Project for the ALS arising out the Genome Project. And actually, this represents the world's largest bioethics program. The European Commission only started funding of HGP when it had set up the ELSI program. Since then, there have been contemporary efforts going on to answer some of bioethical challenges of human genome project. So these 12 points are representing the ELSI. So first is beneficence. Beneficence act of charity, mercy or kindness. The principle of beneficence signifies an obligation to benefit others or to seek their good, and it has been foundation of many codes. The motive of HGP is also based on the principle of beneficence to all. So it should be beneficial for medical purpose, patients, health professional, or agriculture field, or also for our environment. Second issue is do not harm. It is the basis for the principle of justice and confidentiality and philanthropy. Two main ethical arguments in human genome projects revolve around the moral concept of justice and confidentiality. I will discuss these two issues later. Third is human right. In simple terms, the human body cannot be used in an experimental organism without any consent. So, these human right or consent about human right are inducted in the codes governed by the medical experimentation, and these codes are called a Nuremberg Code and Helsinki Declaration. I will discuss about these code and Helsinki Declaration next lecture. Fourth one is animal right. So animal ethics, about animal ethics I have already discussed in previous lecture. Fifth one is authority. So actually, this human genome project is a multidisciplinary project. So this project required multidisciplinary approach involving medical expert, biologist, biostatistics, bioinformatics, a computer expert, and many other for the data banking and analysis. So the multidisciplinary authorization of genome project has one hand rewarded it with speeding the process. On the other hand, it aroused conflict of interest between different groups from the different areas. Sixth concern is autonomy. The concept of autonomy in bioethics gives each individual the recognition of human capacity for self-determinism and being different in spite of sharing same DNA which regard a common heritage. So that is the big issue. Ownership issue, the competition for genome sequence between publicly funded genome project and what's leading private company Clara Genomics. Actually, there was a conflict of ownership between the government institution and this private Clara Genomic company. So the fear behind this is unwarranted risk of data ownership. Eighth one is justice. The other side of the possibility for transforming medicine is who will actually benefit. Will everyone have access to such revolutionized healthcare? Like you can see in developing world, human genome sequencing may not be the first choice for the better treatment. where millions of people do not even have access to basic medical facilities, like in poor countries or tropical countries in or Southeast Asian countries. This HGP cannot be the first choice for the treatment. The ninth is confidentiality and privacy. It is not surprising that in few next few years, the developed countries will be able to get uh, individual genome of every person of or every citizen of their country. So the privacy of genetic information is reinforced not only because of presumed prospects for use of such information beyond medical reason, for instance, discrimination of genetic underclass at different levels. So this is the big issue. Tenth one is the responsibility. The responsibility of use or misuse of genetic information is an individual decision. But what is useful for one may not be useful for others. So that is also a big issue. Scientists and social duty. The scientific community has to bear moral responsibility for using very powerful knowledge as agreed the UNESCO Declaration on Human Genome and Human Rights. But there always lies the danger of misusing scientific freedom. And finally, consequences. The consequences of HGP are affirmatively only when used in proper way for well-being of all humanity. So it has a tremendous potential to develop new drugs. Uh, we have to use precautionary principle approach, which says we need to be very careful to avoid the harm. Actually, Human Genome Organization has proposed guidelines to address some of the ethical and legal requirements related to Human Genome Diversity Project. And finally, HGDP makes us consider our roots and their importance in modern society. So that's all about Human Genome Project and ELSI related to HGP. In next lecture, I will discuss about some other issues. Till then, bye bye. Take care. Thank you.